Okay. All right. Welcome, everybody, for the fourth session in our Stream Team Academy Creek course on an introduction to mussels. We're really glad that you guys can join us this evening. Today, we're going to have Steve McMurray from the Missouri Department of Conservation talk a little bit about mussel diversity and identification. So I hope you all have your shells with you to help kind of identify. Because we can't do this in person, there will be some challenges as far as questions go for identification, but please use the chat box for asking questions, and then our panelists will address them as we go along, and then I can interject for Steve whenever he kind of takes a break in his presentation to address those questions. So Steve comes to us from Columbia. He's been our state malacologist with MDC since 2004. He has his bachelor's and master's from Eastern Kentucky University in environmental resources and applied ecology. He has been wonderful as a resource for stream teams to help identify those mussel shells that we find when we're out and about. Yeah, with that, I'm going to go ahead and turn it over to Steve to get started, and then I will mute my microphone and turn off my camera so that he has the best bandwidth to move forward.
Great, thank you, Amy. And thank all of you guys for tuning in this evening, for listening to us talk about mussel diversity and identification. And so the second part of our presentation on identification is going to be a little tricky. And so I had ended up retooling my presentation a little bit because what I normally do involves me being able to walk around a classroom and point things out to you. And so in the absence of doing that, we're gonna give it a different shot and see where we get. So we're gonna start with diversity. And just a brief introduction. What are we talking about? We're talking about native freshwater mussels. They're also commonly known as bivalves. They have two shells, and each shell is called a valve, so bi meaning two valves. Unionoids, naiads, or clams. And Dr. Barnhart talked about this a little bit in his first presentation on biology. And so what we don't want to do is confuse those with introduced zebra mussels, introduced Asian clams, and our native fingernail clams. I know most, if not all of you, have seen Asian clams in rivers in Missouri, probably native fingernail clams as well. And hopefully you haven't seen zebra mussels, but you probably will before too long. So classification-wise, all of the clams and mussels, both invasive and native, are found in the class Bivalve. And that is split into this group called the lamellibranchia. And they're lamellibranchs because if we cut through the shell long ways, you see the foot here and the outside of the two shells, they have these two gills, lamellibranchs, which are folded so that it looks like there's four, but there's really only two, but they're folded in half. And then we slip down here to the heterodonta, and that splits us into our two orders, the Unionida and the Veneroida. The Veneroids, it's a tough thing to say. That's where Corbicula, Asian clam, comes in. Our sphaeriids, native key clams, and then zebra mussels. And then the Unionida is where all of our native freshwater mussel families occur. Anytime you see a DAE at the end of a word, it designates a family. So what's the difference between Unionida and Veneroida? Having a tough time saying that last name. Both groups brood the larvae in the female gills rather than releasing eggs into the environment which then are then fertilized by male. The Veneroids release either fully formed juveniles, in the case of fingernail clams or Corbicula, Asian clams, or a pelagic veliger larvae, as we see in zebra and quagga mussels. And only the Unionida have a parasitic larvae that requires a vertebrate host. And Dr. Barnhart covered that in his presentation on biology and reproduction. This is a worldwide fauna, again, talking just about our native freshwater mussels. worldwide fauna. Very few land masses on the planet have at least one species. And this is a bit older of a map too, and so I'm pretty sure that the numbers for Africa and South America have gone up a little bit and changed as people are working through those models. And freshwater mussels reach their point of highest diversity, biodiversity in the United States, so in North America. And if we look at this, some of the different shell shapes and species that we see in the Unionida, they're divided into the Unionidae and the Margaritiferidae, which are the only two families that we find here in North America. These other families, Mycetopodids, Arinids, Ariids, are found in different places across the planet. So South America, Australasia, Africa. But the Unionidae and the Margaritiferidae reach their highest diversity in North America, and that's where we're going to focus our efforts. If we zoom in on North America, and using this graph, we can see that the southeastern United States, Kentucky, Tennessee, Alabama, really have the bulk of our biodiversity. And that's true, we see that same pattern for other aquatic faunal groups. Fish, insects, a number of things reach their apex of biodiversity in the southeastern United States. So a little bit more on phylogeny. We're going to break this down a little bit more. So we have the Unionidae and the Margaritiferidae. There's only one genus. These numbers in parentheses are genera, and there's only one genus in the Margaritifera called Margaritifera. And the Unionidae is divided into two subfamilies, ending in NAE, the Amblemini and the Unioninae. And then those go into our tribes. The tribes are important, and we're going to talk more about the tribes because they help us to kind of understand the identification of the animals, because some of the characteristics that are used to classify into the tribes in Odontinae, Quadrilinae, Pluribimini, Amblemini, and Lampsoninae, those are some of the same characteristics that we can use to identify the animals that we have in our hands. So, and as a joke, each of you can try to say these tribe names 15 times really fast without messing them up. So to put it into number of species that we have in North America, we have about 300 species. The last assessment that came out of names was a few years ago, and they had 296 species. But since that time, a couple of things have been redescribed. And not that they're necessarily new species that we've never seen before, but they were kind of lumped into a different name, and genetically they are separate. And so some of that work is ongoing now. And in Missouri, we have about 69 species reported from the state. What we have evidence of shell material that we can actually look at and see is about 65 species. But I learned a couple of weeks ago from Dr. Barnhart that he has found a new species for the state that's never been documented here before. And so once we can all get a chance to look at that shell, and then it'll add to our list. So 66 species. Now, to put that into perspective, if we look at our former alliance with the Big 12, we can see that we were at the top of the Big 12. As we're getting into football season here, and hopefully basketball season will start soon too, our 69 species really put us at the top. Our move to the Southeastern Conference, however, put us at a slight disadvantage, and hopefully, fingers crossed, much like adding some new recruits, we will be able to add some new mussel recruits into the state and bump our numbers up. Try to get up here close to my home school here, University of Kentucky. Why is Missouri so diverse? Why do we have such a diverse fauna? It's really because we are fortunate to be at the intersect of four mussel faunal regions. Excuse me. We have the Upper Mississippi faunal region, which extends from pretty much the Missouri River, a little bit south of the Missouri River confluence, all the way up to the headwaters of the Mississippi River, and even up the Merrimack, Gasconade, and Osage. So it even takes over some of the Missouri River tributaries that are the faunas are really very reminiscent of Mississippi River. The Great Plains here is very depauperate, but we still have a number of unique species that occur in that pretty vast region. The lower Mississippi, where we pick up in the Boot Heel and southeast Missouri, St. Francis and Black River systems, and then the interior highlands that we see here, and specifically the Ozark Faunal region, which we share with our friendly neighbors, Kansas, Arkansas, and Oklahoma. Now, there are several species that are endemic to the Ozarks, and there is one species, the northern broken ray, which is actually endemic to Missouri. It only occurs in north flowing streams in the Ozarks. Okay, so just a pretty brief recap of some of what Dr. Barnhart talked about with life cycle, because this will help us a little bit as we get into talking about the tribes. But spawning is basically the release of eggs. And the females do not spawn into the environment. They actually spawn the eggs from their ovaries into their gills, where they are then fertilized. Those are then brooded inside the gills for a few weeks to a month. And release strategies, they are two brooding strategies, excuse me, bradytictic and tachytictic. Bradytictic are winter brooders, so they are, fertilized in the fall, and they brood those young over the winter and release them in the spring. And then tachytictic are spring, summer, and fall brooders, so they're fertilized in the spring and they retain them throughout the spring, summer, and fall, and they release them in the fall as Glochidia. Now, Glochidia, the larval form of freshwater mussels, can be released as either free Glochidia, where they're just broadcast out into the environment, or they can be packaged up into these packets that are called conglutinates that look like fish prey, basically, something that a fish, a small fish, or even a large bass would eat. And then we also have host extraction, where we have in this situation that you can see here on the right, where this mussel has a lure developed that it does actively flap in the water so that it will attract the fish to bite its gill and get a big mouthful of Glochidia. And then we also have some mussels that actually capture the fish. So for the margaritiferids, and yes, you can think of margarita every time you say that word, because I do. Margaritifera is the genus. We have one species that's found here in Missouri, Margaritifera monodonta, the spectacle case. I know the name says Cumberlandia. That's kind of a tricky thing that you'll run into with mussels if the names change, and they sometimes change fairly regularly. And it is a pain in the neck to kind of keep track of it. Usually, if you can follow either the common name, spectacle case, or the specific name, in this case, monodonta, you can narrow down that Cumberlandia, or that Margaritifera monodonta used to be Cumberlandia. They are spring brooders, and they produce tiny Glochidia and lots of them. And they're often found in these dense aggregations around or under rocks or between big boulders. Moving into our tribes, the first, the Anadontinae. These are species that have thin, smooth shells with very weak hinge teeth, and we'll talk about hinge teeth in just a moment. They have hooked Glochidia, which you can see here, so little pacmen that have these nice, really terror-looking hooks coming out of them. And so they attach to the fins or skin of various host species. They are winter brooders, and they have a spring release. The Lampsolini, which is our most speciose group or tribe, is also the most diverse in terms of how they get their Glochidia onto fish. Most of them spawn in the fall, brood over the winter, and release Glochidia in either spring or summer. Many of them have the mantle lures attached that you see here on the top right to attract in host fish. Some have these elaborate conglutinates, packages of Glochidia and unfertilized eggs that resemble something that a small darter or minnow would eat. Some, like the Epioplasma or snuffbox, actually catch fish. Others broadcast their Glochidia out into the environment and just hope that a host fish swims by. Moving on to the quadrillines. They are spring or summer brooders, and they release conglutinate fragments or fregokidia. They also have very heavy, thick, lumpy shells. You can see one here has these nice lumps on the side of it. They're mainly hosted by catfish or cyprinids, and rather commonly found across Madeira. Amblyomini and Pluribimini, we're going to lump these two together. They are summer brooders. They can release either simple conglutinates or loose lochidia, and there are several, 5 or 6 genera that are included in this. So, in terms of diversity, that kind of takes us to a good place to take a break. We'll have another little spot to take a break as we get into ID. So I'm going to ask Amy to jump back on here and see if there are any questions that I can answer over this first portion, talking about diversity.
So far I'm not seeing any questions, but I'm wondering if maybe my chat might be non-functional. I know my Q&A, I am not able to see much in Q&A except for like a line, but it looks like Jenna asked or answered a question about where we can see these recorded afterward.
Okay.
But yeah, if anybody has any questions, yeah, please use the chat box and we'll address them as we go. But right now, I think we're good.
Okay. All right. Well, we'll just go ahead and jump in. So for mussel identification, this can often be the most frustrating and most rewarding thing that aquatic biologists will do, at least in my opinion. And most people that work with freshwater mussels, if not all of us, would agree with that same thing. So there will be a couple of spots in here where I'm going to ask you questions, and you can just put the answers in the chat box. We're not going to single you out or fail you for not passing the quiz. There will be a couple, so we can call it a quiz tonight. So hopefully you guys all have your thinking caps on. So mussel identification. You can describe it as either variety is the spice of life or an exercise in futility. Both of those apply sometimes to the same species at the same time. So you're presented with shells. And first quiz question is, how many species do you see here? Be honest. It's either one or two. And sometimes there isn't a wrong answer. Oh, I just saw somebody answer one. Very good. Thank you. It's actually two. Mapleleaf and winged mapleleaf. And now I guess, it doesn't matter. It doesn't matter that these are different species. From a biodiversity standpoint, yeah, it matters. It matters that these are separate. But from a legal standpoint, it matters a lot because mapleleaf on the top left is one of the more common mussels, especially in the Ozarks. And then winged mapleleaf is one of the rarest mussels in Missouri, known from really only historically from 4 locations. More recently only from 1, and it's federally listed as endangered. And so handling this species is a no-no, and handling this species is a lot less illegal. So I know most of you, if not all of you, have used dichotomous keys before. They are like the standard for biologists, for naturalists to be able to use to identify things. And there are dichotomous keys for freshwater mussels. This one is in Ron Esch's book, Naiads of Missouri, which if you can find it's out of print. If you can find a copy, you're interested in mussels, I would highly recommend it, mainly because it's got a lot of biological and ecological information in there that you just don't find in other places. And actually, when it was published, Ron, he took and put into print a number of things that mussel people knew, and nobody had ever written it before, and so it was difficult to cite it, but he ended up including a lot of conversations that he had with famous malacologists at the time. So anyway, so he has a key. And as we all know from following a dichotomous key, you have step 1, you have 2 choices. Either shells have knobs, pustules, or parallel ridges on the shell anterior to the posterior ridge, or they don't have those characteristics. And if you follow this down, eventually, if the shell in your hand is Orthoposis siphius, the sheep nose, then you get there. Easy peasy, right? Well, that's not actually as easy as what you think it is for mussels. Dave Strayer and Jerka in 1997, and the Freshwater Mussels of New York pointed this out, that keys based on shell characters are inevitably filled with vague, subjective terms, are frustrating for beginners to use, and you're going to misidentify many shells. And you should know that if you rely solely on a key or two keys, you're going to misidentify many shells. So what are we left with? Well, in the words of the late Tom Waters, curator of malacology at the Ohio State University Museum of Zoology, practice the art of Zen malacology. And I mean some of this tongue in cheek as well. But so what is the art of Zen malacology? Well, the art of Zen malacology, this is a mucket. It's a mucket because, well, we know it's a mucket. I know that's difficult to get, and it sounds almost like I'm being a jerk about it, and I promise I'm not. But you know, there are characteristics that we can use. The shell, it's a nice, thick, heavy shell with sometimes with a raying pattern. Sometimes it has this nice light brown periostracum color. Sometimes it has a white nacre, but in reality, it's because when you picked up this shell, it felt like a mucket and it looked like a mucket, and so it's a mucket. This species is also kind of a grab bag because we often get it confused with several other things, and then finally we're like, oh, it's just a mucket. And I did see a question pop up on my chat. I'm not going to go back to those two, but I am going to go through characteristics that you can use to identify shells. So hopefully some of you took your assignment from Travis to heed, and you actually were able to get out and pick up some shells as we go through some characteristics. So, oh, or just flip a coin. I got ahead of myself there. Really, just flip a coin. Nearly everyone who works with mussels has one of these shells in their office, sitting on a bookshelf or in their lab, or bouncing around on the dashboard of their truck. They have no idea what it is. I don't know what this one is. This one we picked it up actually on a stream team field trip on the Merrimack River, Castlewood State Park. And Karen Couch, amateur malacologist, amateur only in the sense that she doesn't get paid to do it, found it and just brought it up to me. And she's got considerably more experience than I do, and handed me the shell and said, "What is this?" I don't know, Karen, if you don't know. It actually has characteristics of about 3 different species. And so sometimes flipping a coin and making a best guess is the best that you can do. Or ask the experts. Sometimes your experts look like these guys. Sometimes I feel like Mo, other times I feel like Curly. And really, to be honest with you, sometimes the experts don't know. I often remember a story of a colleague who went to the Ohio State University Museum of Zoology. They have one of the largest holdings of freshwater mussel shells and individuals preserved in the world. I think they're upwards of about 70,000 lots. And there's a lot of specimens from Missouri in that collection. And it was the curator of that collection for decades was Dave Stansberry. He was one of the preeminent malacologists of the 50s, 60s, and 70s, and he was one of the godfathers of mussel identification and mussel conservation. And you go into his collection room and you open up one of these drawers, and all of the labels on it say Pluribima spa. He couldn't get it down to species. There's just so much variability there, and he just couldn't do it. And you think, man, if he can't do it, then maybe it's not so bad that I can't do it either. And sometimes we smack each other around when we're in the field too. So in absence of having a tricorder that we can all use to point at the mussel and have it tell us what it is, how do we identify these things? We can't use a key. Sometimes the experts are goofballs, and you don't always know if they're telling you the truth or not. So what do? Well, there are some techniques that you can use. The first question that you will be asked by anybody that you hand a shell to and say, what is this? Or can you help me identify this? Is where did you get it from? Identification by range is often not the best thing to do because, we can get surprised. Things can occur in places that they don't really never been found before. But this is the first question that you will be asked by anybody that you hand a shell to. Say, which river did you get it from? And so that's an important question. And we can use this here, especially for some species, identification by range. And so if I happen to be out and I pick up a shell that looks like this, but I'm in the middle of the Merrimack River Basin, I know it's not this species because this species is only found in the Spring River system of southwest Missouri. And I'm just going to throw in a plug here real quick. These images that you see here are out of the Missouri Field Guide, which we published in 2012. And our field guide and all of the other field guides that are out there to mussels, the shells that are pictured are often not what you're going to see in the field because there's so much variability in shell shape, patterns of coloration that we can't really capture it all. And so we picked the most representative specimen that we could to put into the book. And that's true for any book on mussels. So if you're in the middle of the Merrimack River and you pick up something that looks like this, it's a mullet. And the same thing goes if you're in the Spring River system, in Shoal Creek outside of Joplin, and you pick up something that looks like this, I can guarantee you that after 100 years of people searching there, it is not the mullet. It is neosho. Okay. Going past asking where it came from, there is a divergence of how mussel people handle how they identify things. So I'm gonna run through how I do it. So after I know where you got it from, the next thing that I'm going to ask you about or feel for myself is the shell weight. Is it a thick, heavy shell, or is it very thin and fragile? And that applies whether you have just one of the shells from the animal or both of them. And then the next thing is the outline. The outline shapes that we normally use are here. And the thing that you have to keep in mind, though, is that not all mussels, even the ones that were drawn to represent these shapes. So if we light these up as rhomboidal or round or quadrate or oval or triangular, don't always fit into these exactly. And a lot of older literature and some more recent stuff too will come up with things like subquadrate. So what's the difference between oval and subquadrate? Not much, really. And so it becomes a bit tricky to identify, to use these things, but we try to do our best. So shell shape. If you guys want to throw into the chat box here, how many species do you see here? I'll be able to see it this time or not, but 2. There's actually 1. And that is one of the problems that we see when we use shell shape. This is both the female on the left and the male on the right of Epioblasma curtisi, the Curtis pearly mussel. It's one of our rare and endangered species in the state, but it is a lampsaline, and you may have noticed when we talked about the lampsalini earlier that they do have sexual dimorphism, and so males and females look a little bit different. So it's one thing to keep an eye on and keep watch out for. Now, how many species do you see here? 1, 2, 3, 6. It can't be more than 6. Okay, thank you, Kathy, for answering 3. Actually, what you're seeing here is 1 species, and this is an example of what's called Ortmann's law of stream position. Arnold Ortmann was a famous malacologist in the early 1900s, published a number of papers, and he observed over time that there's a correlation between shell shape and river position. So the same species found in headwaters and small streams is going to be very flat, rough, and compressed. And as you start to move downstream into larger rivers, it's going to be more inflated, smooth, and heavy. And like our good friend Captain Barbosa says, it's really more what you call guidelines and actual rules. But so what we saw there in this picture, we're starting here at the headwaters. So let's say Codaway or Huzzah Creek. And then we flew a little bit farther down, and and we get down lower into the Colorado, and then we go a little bit farther down, and we start to get into the Merrimack. And we start to go farther down the Merrimack, and then finally we get into the Mississippi River. So in our headwater streams, we see these flat, compressed shells, and then moving farther downstream, we see these more inflated, heavy, thicker shells. Okay. So now for identification, we're going to focus on our friend the Mapleleaf here, mainly just because it has a number of characteristics that I can point out to you in one shell. So to begin with, we're going to zoom in on just the outside of the shell. And so, excuse me, the first thing that we'll notice or that we need to look at is our shape, quadrate. Now I'm going to back up real quick. If you had looked briefly at the name of the genus, Quadrula, quadrate shells, even though it does look round, you have to take in the whole totality of the shell. So we have a quadrate shell. And the next thing that will jump out at you, hopefully, is the periostracum. Bivalve shells are made of 3 distinct layers, and the periostracum is the outer, really thin skin layer of the shell. Periostracum coloration can vary from black to yellow to a light tan or darker brown. And oftentimes you can just barely see some here. There are some coloration, there's some coloration that will show up in the periostracum. So how many shells do you see here? How many species do you see here? 1, 2, 3, or 4? Okay, I see some answers coming in. There's actually 1 species here. Excuse me, fawn's foot, or sorry, deer toe. And see, even we get confused on. There's, even though one of the defining characteristics for deer toe is to have this stippled pattern of either upside down W's or almost upside down W's in rows. Usually it's often a narrow row or a narrow ray. Sometimes they don't have any coloration at all. And so you have to, again, go back to your Zen malacology and imagine what it would look like if it had this raying pattern. And also not take, when a field guide says has rays as being set in stone. Some more raying patterns that we see, from large, distinct, thick rays to, almost penciled, stippled rays that go down the shell, rays that have a little breaks in them, or even ones that are wavy. Okay, the next thing that we're going to see on the outside of our shell, and I should have backed up. If you have a shell with you, you can follow along with these as best you can. Not all of these features will be on your shell, but some of them will be. So the next thing is the umbo or the beak up at the very top up there. And the umbo, excuse me, that's the oldest part of the shell. The original shell was laid down around the glochidial, what's left of the glochidial shell when it broke free from its fish host. And so the umbo is the oldest part of the shell. And there's a number of characteristics on the umbo that we can use. But importantly, the umbo is always on the anterior portion of the shell. So it helps us to get our orientations right. And so if we know the umbo is here, that means that we have the anterior side, dorsal is here, posterior is here, and ventral is here. Another feature that helps us out on the outside of the shell is the hinge ligament. That is a piece of, or a portion of the periostracum that grows over and onto the other valve, which would just be sitting right next to this one. And they work together to act as a hinge so that the shell can open and close. Beak sculpturing, sometimes on the beak you can see there might be these really thick furrows that occur in there, or fine lines that are pinched up in the middle. This is called a double loop, or a single loop would just be a round circle. And so you can use beak sculpturing. Another thing is the position and the height of the umbos or the beaks. And so we have the midline of our shells here. So is the umbo closer to the midline of the shell, or is it directed more anterior? And we flipped this around from our last picture. And so The umbo is on this side, so this is the interior end. This is posterior. Height of the umbo. Does the umbo, when viewed from the opposite side, does it go above the hinge line? There's the hinge line drawn in on these, and you can see that on a couple of these it just barely goes above to almost doesn't, and then on this one it does go above the hinge line. Umbo orientation. And so now we have the two valves together here and here, here and here, and we're looking down from above onto the top of the shell, the dorsal part of the shell. So here's our umbos. So do the umbos point towards each other, or do they look like they're facing away, like it's eyes looking forward to see where the mussel is going? The problem with using umbos and the umbono characteristics are that, first of all, that's the oldest part of the shell. And second of all, when the animal is burrowed into the sediment at the bottom of a river, generally anything to the left of where I'm drawing my pointer is what's actually out of the substrate and generally facing upriver. So as water is coming downriver from left to right, it's hitting this. Water has gravel, sand, other particles in it, and so it's slowly eroding away that shell. And so those features are often, especially on older shells, are missing or just entirely gone. Oops. Okay, so going back to our shell here, our friend the Mapleleaf. Next thing that you might notice on these are these bumps. And we kind of divide these into categories. First are pimples, which are really tiny, look like pimples on your face. The next are pustules. Pustules are a little bit bigger than pimples, and they are generally still round, but they can start to approach a teardrop shape. Like you see right here that I'm kind of circling in. The next one down are tubercles. They're even bigger. These can be round, teardrop shaped. This one is almost flat, as you can see here. And then another one that doesn't show up on the shell are knobs. Those are the biggest ones. Think about basically the size of the tip of your pinky finger on an adult shell. You can see some tubercles are popping up over here. And then we have this feature called the sulcus on this shell. Right here, a sulcus is a valley or groove in the shell. And so that sulcus separates this portion of the shell, which is called the disc. Older publications will talk about the disc being free of pustules or the disc having tubercles. And that's what this means. It's just the disc is here. So you have this shallow groove or sulcus that separates the disc from the posterior ridge or slope. Remember our orientation, anterior, posterior. And so on some species, the posterior ridge or slope will be inflated, may or may not have tubercles on it. And then on other species, mostly ones that don't have a sulcus, the posterior ridge or slope, while it's still present, is not inflated, as you see here. Flutings, you can also pick some flutings up here, which are these like line wave-like protuberances that are keep occurring in pattern. We'll zoom in here, we'll go to a different shell where we have some different flutings. So our shell shape here is actually rhomboidal. Cylindrical would be another good word for it, but again, taking in the totality of the shell, it's a rhomboidal shell. And here's our umbo and our beak. You can see that they're pretty tiny on this guy. And then that gives us our orientation, so anterior, posterior, dorsal, and ventral. And so if this is anterior, here's our posterior ridge and slope. You can see that it's really not inflated beyond the rest of the shell. And just posterior to that are flutings. You can see what we mean by flutings here. They look almost like waves rolling in. Another shell feature that we can use here, you guys think about this for a moment and tell me what shell shape you will assign to this. If you want to put that in the chat, that'd be great. And if not, we can jump on ahead. Now, if you're just using this portion of the shell, you might say oval. And actually what this is, because you have to take in the totality of the shell, so all of it, all the way around here, would actually still be a rectangular shell. Umbo and beak, again, gives us our orientation. And then there's this structure called the wing. There are a number of species that have these, that have a wing developed on the shell. And this is an extension of the periostracum that actually goes over and grows over the shell. A group called the heel splitters have these. If you can imagine, this portion, from this portion down, is actually what is would be buried into the sediment. And this thing, which often can and does go up here and make a point, would be sticking out. And so if you're walking around in a stream barefoot, you're going to split your heel on a heel splitter. These come in a wide variety of shapes and sizes, and on some species they even have an interior heel splitter, not only posterior, but they have an interior one as well. However, these do get broken off, and I know you probably saw from the previous slide that it was broken and kind of rough looking. So they do tend to get broken off, whether somebody stepped on it or it just wore away or eroded. They do tend to get broken off. Okay, so before we jump into the interior part of the shell, I'll go ahead and take a quick break here and see if there are any questions just on the external shell features.
Sure. Well, actually, we have a couple of questions that kind of move backwards from just the identification parts of the shell. But first question I saw come up was from Linda Williams, who asked, is the coloration and patterning consistent in one stream area, or are they kind of all over the place as individuals? So if you're in one area, are you going to see different patterns on the same species?
They're all over the place. You can be at the same mussel bed in a river and pull up 10 individuals of the same species and have 10 different patterns of coloration on the outside of the shell. So it's really, most of us that work with mussels over the years have just looked at a lot of different shells, and so we just picked up on those patterns and just have our own little 3x5 index card file in our brain that we flip back through and try to identify those. So
that makes your job so much fun, doesn't it?
Okay. Yeah.
And one more question. Does the mussel select its habitat or does its host?
Oh, that's actually a very good question. And it's almost like a which came first, chicken or the egg. So after the Glochidia attach to the host, which that's, they don't utilize anything, any of the tissue from the host, because they clamp down on the gills or the fins of the fish, and there's just a minuscule amount of fish tissue that's inside of that Glochidia shell. They do dissolve it, but it doesn't really provide them with with much nutrition, and so they do transform from the Glochidial stage into the juvenile stage when they're inside a cyst, and then they drop off. And so really they're where their habitat is and where they choose to live is dictated more by their host than anything else. And so if you have a species that prefers a particular type of habitat and their host fish just happens to be swimming somewhere else because of a flood or a drought, and they drop off, they may or may not be able to adapt to that habitat. Most of our species luckily have a wide range of microhabitats that they can utilize, and so it doesn't always hurt them too much. But no, usually the host chooses the habitat, and then once an individual, though, if they can handle being in the substrate, they can move. They don't move very fast or very far, but they can move, and so they will move themselves into more preferred habitat. So, okay. So we'll move into the inside of the shell. And so the first thing that I want to point out again is our umbo or our beak up here at the top. Let's get our orientations again. Anterior, dorsal, posterior, ventral. Then the next thing that everybody notices is the nacre, which is this lustrous mother of pearl layer on the inside of the shell. And mother of pearl, this is where pearls come from. And so the same material that lays down the inside of the shell is what makes pearls. It's very smooth, almost silky smooth layer. It's very thin, not as thin as a periostracum, but it's very thin. And then and nacre colors sometimes can be used to help us out. And so how many species do you see here? 1, 2, 4, 6. See if anybody bites and puts a number in. Actually, there's 2 species here. One on top, all 4 of these, commonly known as a spike or ladyfinger, has the usual coloration pattern is to see this almost dark royal purple. But we can have white ones, we can have ones that have salmon, we can have ones that have a mixture of purple and salmon, and we can find individuals with these color patterns in the same bed, generally right next to each other. So it can be a bit tricky. It's often tricky, it's made trickier when you're in streams in Missouri in the southern part of the state that have a species known as Washita kidney shell that have a lustrous white nacre and look from outline, weight, and lack of any other really readily identifiable features on the outside of the shell, look exactly like spike. And so it does make things kind of fun. Down at the bottom, we have deer toe. And so just another example how you can have both white and pink coloration at the same time. There are some species that do have like a dark purple color, purple warty back. And once you see that, you can go, oh, yep, that's purple warty back. The same thing for pink gill splitters, which generally, usually, always have a pink nacre. Mussel people like to use the word generally and usually a lot because we can't say beyond the shadow of a doubt that something will always have purple nacre or white nacre or pink. Okay. Other structures that are on the inside of the shell that are important are teeth. They're not teeth used for chewing, mastication. They work in conjunction with the hinge ligament to provide an axis that the shell can open. And so we have cardinal or pseudocardinal teeth here, which are big, usually big, thick, heavy, triangular shaped teeth. And then lateral teeth, which are more refined and finer edged. And depending on which side you have, whether you have the left or right valve, will have either 1 or 2 teeth because they nest in together and act as part of the hinge for opening the shell. So I should have pointed out earlier, and I neglected to, but since we know that this is the umbo and this side to the right is anterior and this is posterior, that allows us then to, if we can kind of turn the shell a little bit so its anterior is our anterior, so our chest, our front, then what we're looking at here is the inside of the left valve, and then what we were looking at before was the outside of the right valve. So the teeth are separated by this structure called the interdentum, and it will either be smooth or have some serrations or be knobby on its own. And then another feature underneath of the interdentum is the beak cavity. And depending on which species you're looking at, the beak cavity will be very shallow, or it may be deep enough that if the shell is big enough, you can actually put your pinky finger in there and lose the tip of your pinky finger inside the beak cavity. Now, looking at teeth, we can go from our anodontines that we see up here, which, remember when we talked about our tribes, have little to no teeth. They're actually very reduced. There is a cardinal tooth there, and this slight swelling would be what would be the lateral tooth in this species. And so they're pretty much non-existent. And you can also see that there is no beak cavity here because there are no teeth. There's no interdentum here because there are no teeth. And then we move down where we see this formation of starting to get more triangular shaped teeth with a very narrow interdentum between the cardinal or pseudocardinal teeth and the lateral teeth. And down here on the bottom right, we have a very thick, heavy, knobby pseudocardinal tooth. and a very well-defined interdentum before we go into this very short and stocky lateral tooth. And I know it's difficult from a picture on screen, but you could, if you were able to, you could stick your pinky finger in this beak cavity right here, and it would go all the way back to there.
Hey, Steve, and don't forget
Yes.
to tell us what kind of mussels those are.
Oh, yes. Okay, I'm sorry. Yeah, yeah, I wasn't even thinking about that. So on the top here, this is the floater. In your field guide, if you're looking at the Missouri field guide, it will be Utterbachia imbecilis. Had to remember which genus name we used for it because it's changed since then. And then this one, I believe, is a broken ray. So one of the Lampsless Reviana complex species for broken ray. And this is a purple warty dot. The outside of the shell is just dotted with, it's covered with pustules and pimples, and it's a very flat shell. But this is one that I can go say always has this dark royal purple coloration. Never seen it with anything else. Okay. And so here's some more teeth. And what we're looking at on this picture is this, the lateral teeth. And so you can see how we have our hinge ligament here, right there, and the lateral teeth are actually starting to go down. So anterior, posterior, so posteriorly they start to drop off and point towards the bottom of the shell. This is spike here, or ladyfinger. You can see that we have a pink nacre on this one. And then this shell here, which I believe is three-horn warty duck, you have these nice, thick, heavy shells, but then the lateral teeth are very short, whereas these are long. The lateral teeth are short, stubby, and they are, I know the bottom of the shell is on here, but they are parallel to the ventral margin of the shell. Okay, really that's it. Those are the features that we use every day, every time we're out to identify these things. And so using our field guide here, this is a Mapleleaf on page 79 of the field guide. I'm just going to run through it real quick. So pick this shell up in the Merrimack River for our example's sake here. So I know that Mapleleaf is a possibility to occur there. And then what we're really looking at here is this written shell description. The shell is moderately thick to heavy. They are. They're very thick shells, even for their size. This one is only 3 inches, from anterior to posterior, but still that would be a very thick, relatively heavy shell. It's quadrate. It is compressed to inflated. Compressed means if you have the two valves together and you're looking at it from on top, dorsally, is it thin or is it expanded or inflated? And so compressed would mean that it's a very thin shell. And that, but again, that depends on the habitat, and they tend to be more inflated and softer substrates. The beak sculpture is similar to the adult shell. You can see we have these dots that just start going up, and those, and I know from experience, continue all the way up onto the umbo. 2 divergent rows of pustules from umbo to ventral margin, right there and there, often separated by a sulcus. Pseudocardinal and lateral teeth are distinct and massive. They are. Periostracum is greenish to brown, often with green rays, maybe lacking, and the nacre is pearly white. So those really are the characteristics that we use. And then we went ahead in our field guide, and others do this as well, to give you similar species. So you can flip through and say, well, maybe it's a winged Mapleleaf. And this really like picture pages more so than anything else. Most of us that work with mussels have about 10 to 15 to 20 different field guides or mussel books on hand, and we flip through all the pictures of them and we make a good guess most of the time. So variety is the spice of life, and that's what we can definitely see in this picture. So anybody want to guess how many mussels or how many different species are in this picture? Okay, I'm not seeing any takers on that one. These are actually all Mapleleaf, the one that we just worked through identifying. And these are in the Lamine River from the same bed. We found all of these individuals, and we moved from smooth ones without any pustules or any of the characteristics really that we used to identify, except for the outline of the shell. And they look very similar to ones that we can identify. So we'll zoom in. This is a bit blurry, but we'll zoom in. You can see we've just barely got some pustules developing here, but we do have a sulcus. And we have a sulcus here, and we can see that there used to be pustules or tubercles up here. And then we get into what is more a more common shape and outline and shell characteristics of maple. Here's our exercise in futility. How many mussels do you see here? And I can tell you right now that there are no wrong answers for this one, especially. Well, I don't know the answer to the question either, to be honest with you. We collected these mussels were collected from the Black River, and, and myself and colleagues Scott Feynman and Andy Roberts, and Andy came back from we went to a bed and we kind of went our separate ways in the river and snorkeled and picked up mussels. And Andy came back and he's like, "Wow, I found these really great ebony shell." And Scott and I said, "No, no, you didn't." And he said, "Yeah, I did. Yeah, I found some." "No, you didn't." And so he pulled up pulled out these shells on the right and said, "Yep, see, those are ebony shell." No, they're not, Andy. And so we sorted them out, and I took this picture and sent this picture to colleagues in Arkansas, and because Black River was in Arkansas, I said, "Hey, can you guys look at these?" And they're like, "Yeah, um, those guys on the right, I don't know, they could just be really big Wabash Pigtoe, but then, it's really cool. The guys on the left, you got some Plurabenas thrown in there, and then, I think the ones in the middle are all Fuscanaya, but I don't know what the species is." And so I sent this picture to a colleague and a friend who works mainly east of the Mississippi River, spent his entire career in Kentucky and Florida and North Carolina, and sent it to him. And he's like, "Man, those mussels on the left, those are some really great ebony shell that you got there." I bring up that story, and I tell you that story because those of us that have been doing this for 30 years, 40 years, 50 years, sometimes we can't even agree on what these things are. So how are people who are just starting supposed to? And the main thing is to, if you identify one and you get it wrong, there's nothing wrong with that because we get them wrong all the time. I still get confused on common things. My assistant Scott brought back a shell that he was very excited about finding one time, and he brought back the shell, and periodically he would look at it, and then one day a few weeks later he came up and he's like, it's pond horn. I don't know why I didn't get that when I was in the field. It's a very common mussel, and so it's just we all get perplexed from time to time. So there's our expert panel pondering the pigtoe question. That picture that I just showed you they were all pigtoes, and they give those of us who work with mussels on an everyday basis fits. And there are a lot of opinions out there on what they are. And so it's really a dilemma that we're trying to work through with genetics and some morphometrics on the shells, but we're just not quite there yet. So stay tuned, and maybe we'll be able to answer that question next year. Okay, but now I brought up a few books. There are a number of useful books and field guides that will be quite helpful to you in your journey. The first one, to toot our own horn here, is A Guide to Freshwater Mussels that myself and Travis and Andy and a few other people authored and pulled together. This publication is freely available from the department. You can contact our publications people, and they're free to residents of Missouri. You can also contact your nearest conservation department office. Many of them have copies on hand that they're willing to give out. And if that doesn't,
I added to the chat that they can contact us at streamteam@mdc.mo.gov and request a copy, and we'll be happy to get those sent out.
Okay, great. Thank you. And if you are new age kind of person and you want to have an electronic copy of it, we have a PDF version of it that we can send out to you as well. I still like to have the book in hand, but PDFs work pretty good too. Missouri Naiads, A Guide to the Mussels of Missouri by Ron Esch. This was first published in 1984 and then updated in 1995. It's out of print. There are no more copies available. But you can find them used on Amazon. I know they're there. Actually, you can find used copies of this one on there too. I actually found a copy they wanted $20 for on Amazon because it was signed by the author. And I looked at the cover and I actually signed it for someone sometime. And I forget the name, but it was, dear so-and-so, thank you for your interest in mussels. And so, so anyway, so you can buy one for $20 or you can contact us and we'll get you one for free. I'll sign it too if you want. But this will be a good reference to have if you're really interested in mussels and not just in identification, but also biology and natural history and ecology, because Ron put a wealth of information in this book, which is sometimes overlooked. And I do have this as a PDF that I can send out, but I may have to compress it before I do. It's a pretty hefty book. Surrounding States, Illustrated Guide to the Unionida Mussels of Kansas. I don't know if Karen has any more copies of this available. Karen Couch did this, and it's illustrated because she drew and painted all of the plates that are in there for all of the different mussels of Kansas. And it's really beautifully done publication. It's self-published. It's spiral bound, and so but Karen's a wonderful person and a great malacologist. And then Field Guide to Freshwater Mussels of the Midwest. This is published by Illinois Natural History Survey. This was one of when it came out, everybody was scrambling to get a copy of this, and they still have them for sale. I think they're 12 or 15 bucks if you contact the Natural History Survey. The thing that you have to be careful about is that they did not include Ozark species in this book because Ron Esch's book was out and already covered. And so those are missing from that. So if you're working in the James River Basin or the Spring River Basin or the Current River Basin, the species you're finding in there are not going to show up in that field guide. But if you're working in the Mississippi River or the Merrimack or North Missouri, then they'll be there. Freshwater mussels of Tennessee, for those of you that might be down in the boot heel, and the freshwater mussels of Kentucky, both of which have a lot of nice, really good pictures on the different mussel species that occur in those states that we sometimes share with them. And so if you're down in Cape Girardeau or Kennett or even south of St. Louis, you're close enough to Kentucky and Tennessee that may actually look like some of the specimens that they photographed and put in those books. Okay. So I'd just like to close by thanking all of you for your time this evening. And I know that covering identification of these animals probably is not the best way to do this over WebEx, but we wanted to at least give you some information and at least get you started down this road. It's challenging, but hopefully it's also enjoyable once you get to do it. My email address is here. If you have a nice picture of a shell that you found somewhere and you want to know what it is, or know if your identification is right, send it to me. I may not be able to get to it right away, but I'd be happy to look at it. And if the picture is not clear enough, I may have to beg off and tell you that you might have to send me a better picture. But if you've got enough characteristics in there that I can use, then I'd be happy to identify it for you. And then I just want to put a plug in for Travis's presentation on Friday morning at 11 a.m. Travis is going to talk about large river mussel issues and mussel outreach opportunities. And so his will be the last presentation in our Stream Team mussel workshop series that we're doing here. So please tune in to listen to Travis. Thank you very much.
Excellent. Excellent. Thank you so much, Steve. And we do have some questions, so I'm actually going to back up about half an hour here.
Okay.
Let's see. And I think it's just a few. So, but Elliot Lipin was asking, do the same identification traits stay consistent regardless of mussel age? So is there an influence on how old the mussel is on the traits that it has in order to identify it?
Yes and no. The no part comes from if it's a feature like the umbonal characteristics, then that's going to be generally be eroded away in older shells. But a young, let me back up here just a few slides. Okay, these are all Mapleleaf, and this is a younger specimen, as is this, and these are older. And you can see that even these older specimens still retain at least some of the characteristics that we use to identify our Mapleleaf. One trick that Dr. Barnhart talks about using, especially for the pigtoes, this is a nice mess of pigtoes here. They're in two genera, Fusconaya and Floribima, but they're commonly, their very common name is the pigtoes, Wabash pigtoe, round pigtoe, Ohio pigtoe. And so we just kind of lump them all together as pigtoes. But these darker lines that are on the shelves, these are their growth rates. They're not necessarily always annual. They represent rest periods, somewhat analogous to an annual growth, but not always. But you can look at in order to identify some of these, you can look at these younger shells. So just imagine that from there all the way there is just gone, and you're looking at this younger shell up here. And that will often be the best thing you can do to identify these. And no, I still don't know what they are.
Okay, let's see. Yeah, Ron Brooks made a really good point in that the variety and variation almost defies the rule of genus and species. They kind of seem to be in violation of the rules of inherited characteristics. And yeah, and I had responded and said, yeah, I guess it just takes a trained eye on those particular features that you had highlighted earlier in the biology or in the anatomy of the shells. Does it occur in other species? And I would probably say yes, it probably occurs in most species.
Yeah, I think it does. And I mean, speaking of like me personally, I am terrible at identifying wildflowers. I've got like 5 or 10 that I like, and I know what they are, and I can always pick them out. But my kids are always saying, "Hey, what's that pretty flower?" I'm like, "Oh, I don't know what it is. How much is it? I'm looking a river." It really is. It's just over time developing an eye for things, and just having my little CPU going on in the back of my head that is constantly analyzing what I'm looking at. But the other thing to keep in mind is that mussels are famous uh, ecomorphic animals. And so what that means is that oftentimes their shell characteristics are defined by their habitat. And so, but the thing is that let's say, for example, that these 7 here, actually, go ahead and say that these 11 animals right here, these 11 individuals are all the same species. The thing is you're looking at a very plastic feature, the shell, to be able to identify them, when in reality, the actual animal on the inside of the shell looks exactly the same in all 11 of these. And so that's the trick that the issue that you run into. You're using a very plastic feature, the shell, even though it's nice and hard and can make that noise. It's still very plastic, and it just changes with the environment.
And I guess that kind of leads into the next question that came from Georgianne Bowman. When you are in the field, would you actually open a mussel up to look inside, and would that harm them?
You can. Some close up a lot tighter than others do. These guys that are up on the picture right now are pretty famous for once they close their shell, good luck getting it back open again. But you do have to be careful. Let's see, I'll back up here a little bit and go into the question. Forgive me. Oh, right here. So we talked about the hinge ligament and the teeth acting as hinges that allow the shell to open up and close. And right here, this is an adductor muscle scar in SCLE. And then there's another one over here. So there's one anterior and one posterior. And in order to close the shell, they contract that muscle and it closes the shell down and they hold it closed. And then when they want to open it, they just relax those muscles. And then the hinge ligament is what actually starts to force it to come open. So you can open them up a little bit, but you do, and you get a feeling for this when you're handling muscles. But you reach a point where you snap one of those adductor muscles, and then basically your best bet is just to preserve that animal because it's going to be a goner because it can't really close its shell all the way. But you can open them up slightly, maybe a quarter of an inch, at most an eighth of an inch, and that's enough to pry it open and then use your fingernail or a small tiny stick, and you can push the tissue back just a little bit from the shell, and you can see the nacre color on the inside.
Okay, and that actually kind of leads into the next question: is how often do you need to actually kill the mussels to identify them or attempt to identify them?
Rarely. We try not to. It does happen sometimes. And for some genetics work, because the proof is in the pudding. So for some genetics work, those researchers like to have the whole animal so that they can have genetics, DNA, but they can also have soft tissues that they can use for comparative purposes. And then they also have the shell, and they can link those three things all together. And we have, preserved some animals, some of the Pigtoes, because we had a project going with some colleagues in Arkansas and Kentucky to try to figure out what Pigtoes we were finding in different places, and so we did end up preserving animals there. But generally, we do our ultimate very best to not actually kill the live animals to try to identify. Oftentimes there's, depending on your field crew, there's 40 to 50 years of experience out there, and you just kind of use your knack and bring your boxing gloves to be able to argue with your partner over what species you have. So.
Yeah, fair enough. Just two more questions, and I think we're good to go. So is there as much variety in oysters?
I don't know. Honestly, I don't. I think that there is, but I know I do pay attention to some of the listservs that the amateur shell people belong to, and they do a lot with marine shells, marine animals, and so they often talk about some of the same problems that we do with identification. So all of our saltwater cousins of freshwater mussels, just that there's a lot of variability in shell features across the board in mollusks, whether we're talking about freshwater bivalves, saltwater bivalves, snails, octopus. There's just a ton of variability that happens with the shell.
Right. Okay. And then I think, okay, maybe one more after that. Are you currently using genetics to confirm the species identification?
Not so much. We have had one project with Arkansas State University and Eastern Kentucky University where it was a blind sampling. And so we would collect the specimens and send them to a colleague in Arkansas who would take tissue out and then retain the shell and the rest of the information, where it came from, all those different things. And so our colleague in Kentucky only got a small snippet of tissue with a number attached to it, and then he ran some genetics. And so we've done some of that, and really the results of that were still very much up in the air. It didn't really help us answer too many questions, unfortunately. But and so as a general rule, we don't take a lot of DNA for identification purposes, but we do use DNA for a lot of more population genetics work. And so helping us to get a better handle on the relatedness between populations of a species that occurs in different river basins. And most of that falls back into artificial propagation work. And so, if we have a species that occurs in the Osage, the Merrimack, and the Gasconade, two of those are tributaries to the Missouri River. All of them are tributaries to the Mississippi River, but they've been separated for millions of years. How do we, can we mix stocks between those three rivers? And so we use genetics work to get a better handle on what the genetic makeup of our broodstock, possible broodstock sources look like. So we use genetics for that. There are people that, there's genetics work that's going on right now, looking at some of the different mussel species that are out there. One is the Gulf maple leaf, which at least presently in Missouri only occurs in the Osage River system, in the Osage River main stem. And it was historically in the 1900s was one of the most common mussels there, but that name kind of fell out of usage for that species. It never really, nobody ever wrote an article that said this is not a real species, don't use this name anymore. Just people stopped using it because it's so very similar to the maple leaf. And so, so people stopped using that name until the late 90s when it started being used again. But researchers have started to take genetic tissue material from some of those individuals so that they can then analyze that in comparison with what other people are calling Gulf maple leaf from across the country, and then we can hopefully come up with a better refinement of what the shell characteristics are that are identifying that species. So.
Great. Okay. One last question. Well, one comment first, which I thought was kind of fitting. It kind of sounds like using hair color and curl to identify a person. So I thought that was kind of fitting.
Yeah.
For sure. And then the last question. Back on one of your first slides, what was the difference between the maple leaf and the winged maple leaf? Yeah. At the very beginning there.
Excuse the high speed clicking here. There we go. Okay. So for maple leaf and winged maple leaf, the the biggest thing is that in maple leaf you have a smooth sulcus. In winged maple leaf, you can see that we have a few pustules that are coming in there. But the other thing is this very straight line, the straight margin right here. And it's there, but it's not as straight and across as on maple leaf as it is winged maple leaf. We also see some rain pattern in maple leaf that you do not see in winged maple leaf. You have more teardropped tubercles, teardrop and flat tubercles that you see here. And then this picture doesn't really do it justice. It's because of the orientation of the left valve. But this lateral tooth on winged maple leaf is very painfully straight. And it's not so much in maple leaf as it starts to drop off. And you can kind of tell too if you draw a box around these. This really does approach way more of a quadrate shape and outline than maple leaf does. And then last but not least, and we can't really do it here, is if you put the two valves together and you look at it from the top, so look at it on its dorsal margin, and you have these two side by side, these are very compressed and these are very inflated. And so that's the other thing that we use. And one last feature that I forgot about, the fluting. Fluting here on maple leaf is very weakly defined, and on winged maple leaf, it is very, very well defined fluting. Oh, and for those of you that are still tuning in, I forgot to mention this on nacre color. That is a stain. Stains are just due to environmental contaminants that are out there and are not part of the coloration. And so stains can be very prominent. They could be throughout the nacre. They can almost cover it up, or they can just be a few blotches, or you don't get any, and it's just very lustrous white show.
Great. Okay. Thank you so much, Steve. It sounds like your dogs are kind of wanting some attention.
I don't know what's going on with them.
Again, thank you everybody for joining us this evening and spending your evening with us. If you have the opportunity, join us on Friday morning for Travis Moore's presentation on outreach opportunities in large river species. So that should be really interesting, especially for those who are interested in larger river issues. But again, of course, all of these are being recorded and posted on the Stream Team YouTube page. So again, once again, thank you, Steve. Really appreciate the information. Yeah, please email us at streamteam.mdc.mo.gov for the Missouri Mussel book. If you'd like a copy of that, we do have plenty of copies available for the general public. And if you're not already part of the Stream Team program, please consider joining us. So with that, I will go ahead and hit stop recording, and you all have a wonderful evening.
Thank you very much, everybody, for joining us. Appreciate it.